Recombinant DNA technology can be used in the production of human insulin. Now, let's take a look at the production process. First, DNA encoding human insulin is obtained from a human pancreatic cell. Then, a restriction enzyme is used to cut the DNA. Meanwhile, a plasmid with antibiotic resistance genes is obtained from a bacterium. The plasmid is cut open, using the same restriction enzyme. When the DNA fragments and the open plasmids are mixed together, some of the plasmids bind with the DNA fragments, and with the help of DNA ligase, they are joined together, forming recombinant plasmids. Note that some open plasmids do not pick up the DNA fragments and join by themselves. They are called non-recombinant plasmids. The plasmids are then mixed with the bacteria E. coli. This is to introduce the plasmids into the host cells. However, only some bacteria actually pick up the plasmids. These bacteria are said to be transformed, and only some of them have the recombinant plasmid. The remaining transformed bacteria have the non-recombinant plasmid. Meanwhile, some bacteria do not pick up any plasmids at all. In this example, the plasmid we use carry a resistance gene for the antibiotic ampicillin. So, to select the transformed bacteria, we first culture the bacteria on an agar plate containing ampicillin. The ampicillin resistance gene in the plasmid enables the transformed bacteria to survive. They then divide and form colonies. The bacteria that didn't pick up any plasmid cannot survive. After that, we transfer a portion of bacteria from each colony to a second agar plate containing another antibiotic tetracycline. As the transformed bacteria with a non-recombinant plasmid carry an intact tetracycline resistance gene, they survive and divide to form colonies. On the other hand, in the transformed bacteria with a recombinant plasmid, the DNA fragment inserted made the tetracycline resistance gene non-functional. Therefore, the bacteria cannot survive on the plate. From the results of these two agar plates, we can identify the colony formed by the transformed bacteria with a recombinant plasmid, and select these bacteria for further processing. The transformed bacteria with a recombinant plasmid are then cultured in fermenters. In each bacterium, the recombinant plasmid replicates independently of the bacterial chromosome. When the bacterium divides, the plasmid is also copied to the daughter cells. As a result, many copies of the DNA encoding human insulin are produced. Finally, gene expression is induced in the bacteria to produce polypeptides. The polypeptides are extracted from the bacteria and processed into functional human insulin.